Hi class, should we wait for 2-3 minutes? We'll wait for 2-3 minutes more. Okay.
Yes, ma'am.
Can we start now? Is it audible?
Yes, ma'am.
Okay, great. So can we revise what we did last week? So we talked about the structure of a flower, that is gynoecium. Okay, it is what? A female reproductive part. And prior to the last Saturday, we talked about androecium, the male reproductive part of a flower. And in gynoecium, single pistil जो है, that is called as monocarpellary. More than one pistil, multicarpellary. Pistil matlab pistil includes what? 3 parts it has. Pistil has stigma, style, and ovary. Okay. And in multicarpellary, pistils may be fused together or free. Which are fused, it is called as syncarpal. The example is the red one. Example is papaver example. See the fused, the pistils are fused. And in Michelia, it is apocarpus, which means they are free. Pistils are free. See, alag-alag dikh raha hai na humko. So the carpels, we can see it has free. Carpels of pistil. These are free. They are talking about stigma, style, and ovary. And next, inside the ovary, it is saying there is a locule in which placenta is located. And from placenta, ovules come. Ovules के दूसरे नाम है megasporangia. Okay. And we discussed about the ovule. Ovule के structure क्या है? We discussed किया, remember? ठीक है, दिस इज व्हाट स्ट्रक्चर ऑफ ओवरी। पहला के अंदर क्या है, एकदम अंदर एम्ब्रियो सैक है, उसके कवरिंग, उसके बाहर क्या होता है, न्यूक्लियस करके एक चीज होता है विच प्रोवाइड्स फूड। उससे भी बाहर क्या होता है, इंटेग्यूमेंट्स, इनर इंटेग्यूमेंट्स एंड आउटर इंटेग्यूमेंट्स। और इंटेग्यूमेंट्स के बाहर क्या होता है, ओव्यूल, और ओव्यूल के बाहर क्या होता है, ओवरी। एंड एम्ब्रियो सैक इज नॉट जस्ट लाइक दैट, ऐसे सर्कल के अंदर सर्कल है, but how it is attached, it is not in the air, it is attached to a thing called as stalk, and that stalk is called as funicle, and it is not directly attached like this. एक एक एंगल फॉर्मेशन होता है, एक कर्व होता है, वो कर्व मेक्स अ एंगल, वो एंगल को क्या बोलते हैं, हीलियम। ठीक है, और फ्यूनिकल टू द deviation part, it is called as raphe. And the male gametes enter where, from micropyle. दिस इज द माइक्रोपाइल, और जो भी माइक्रोपाइल के ऑपोजिट एरिया को क्या बोलते हैं, चलाजा। ओके, एक्चुअली यहाँ एक स्पेस होना चाहिए सो दैट वी कैन डेमोन्स्ट्रेट टू शो द मेल गैमेट्स एंटर फ्रॉम हियर। ठीक है, अंडरस्टूड सो फार? अंडरस्टूड? डू यू वांट मी टू रिपीट? Yes, class, do you want me to repeat?
No, ma'am.
No, ma'am.
No. ठीक है,
No,
ठीक है,
ma'am.
okay, okay, okay. And then we are saying the mega mother cell, that is MMC, is always a diploid. Diploid matlab 2n. This diploid always will undergo meiosis and forms a linear tetrad. This is a pictorial thing which I have mentioned. This is a mega mother cell. वो हमेशा diploid होता है, that is 2n. And whatever it is 2n, it will undergo meiosis. And it will form a linear tetrad. 4 सेल दिख रहा है इसमें, उसमें से माइक्रोपेलर एंड के 3 सेल डीजेनरेट है, और जो चैलाजल के एरिया में जो सेल बचा हुआ है, that will be what, functional one. और ये जो है that is n, ये भी एक ही बचा है ना, एक ही n है ना, and from that there will be mitosis. Mitosis क्या है, mitosis 1, it will form 2 identical daughter cell. So mitosis 1 से 2 daughter cell हुआ, mitosis 2 से कितना हुआ, एक्स्ट्रा 4 हो गए, mitosis 3 से कितना, 8 हो गए, 2 4 8 डबल हो रहा है, mitosis 1, mitosis 2, mitosis 3, and after mitosis 3 ऊपर के 4 और नीचे 4 है, ऊपर के 4 में से एक बीच में आएगा, नीचे के 4 में से एक बीच में आएगा, सो ऊपर अभी कितना हो बचा, 3, बीच में 2, एंड लुक एट ओवर हियर द नेक्स्ट पिक्चर, ऊपर 3 बचे, बीच में 2 बचे, और नीचे 3 बचे। ठीक है, और अब क्या है, ऊपर के 3 को क्या बोलते हैं, चैलाजल एंड में जो है उसको क्या बोलते हैं, एंटीपोडल सेल्स। नीचे के क्या, अब बीच में जो वो है, दैट इज कॉल्ड एज पोलर एंड। ठीक है, और इसमें नीचे जो माइक्रोपाइलर एंड में क्या है, एक एग रहेगा और दो सिनर्जिट्स रहेंगे। ठीक है, इसमें क्या है, फिंगर लाइक स्ट्रक्चर रहेंगे सिनर्जिट्स में, एंड यहाँ से ये जो है, दिस इज व्हाट, pollen tube. For what this pollen tube is there? For gametes to enter into the ovary. ठीक है, सो इसको क्या बोलिए, मोनोस्पोरिक थे, एमएमसी पहला मोनोस्पोरिक थे, एंड नाउ इट इज 7 सेल 8 न्यूक्लिएटेड। 7 सेल मतलब 7 सेल है और 8 न्यूक्लियस है, 1 2 3 4 5 6 7 8 न्यूक्लियस है, और ये जो बीच का एक है, ये दिस इज 1 सेल विथ 2 न्यूक्लियस, सो इट इज व्हाट, 7 सेल 8 न्यूक्लिएटेड हो गए। ठीक है, एंड इट वाज This name was given by whom? S.G. Navas-Chin. यहाँ ब्रैकेट में लिखा हुआ है, and in your textbooks you will find it S.G. Navas-Chin. ये हो गए. This is the formation of embryo sac or gametophyte. जो मैंने बोला वो रिटर्न में यहाँ लिखा हुआ है. And next is types of ovules we discussed. We discussed about types of ovules. क्या क्या थे? Orthotropous means straight line, 180 degree. जहाँ micropylar end, chalazal end and funicle is in a straight line, 180 degree होता है. Next is hemitropous, which means what? 90 degree formation. Micropylar end, chalaza और funicle, ये देखो ऐसा 90 degree में होता है. Hemitropous. And anatropous मतलब माइक्रोपाइलर एंड जो है, ये जो हार्ट शेप दिख रहा है, हार्ट शेप, ये जो कर्व दिख रहा है, वो पूरा नीचे, एकदम नीचे जाएगा तो वो एनाट्रोपस बोलते हैं. एंड द फोर्थ वन इज एम्फिट्रोपस, वेयर बोथ एम्ब्रियो सैक एंड ओव्यूल आर ऑन द सेम डायरेक्शन. ठीक है, बाहर और अंदर की चीजें एक ही डायरेक्शन में है, उसको क्या बोलते हैं? एम्फिट्रोपस. द फिफ्थ वन इज सिरसिनोट्रोपस, सर्कुलर कॉइल मोशन में जो है सिरसिनोट्रोपस. The last one is Camphitropus, मतलब it is in a diagonal shape, micropylar और ये chalazal end है यहाँ opposite में, ठीक है, but it's in a diagonal form. It's not either left or right में नहीं, it is in a diagonal shape. These are the types of ovules. ये types of ovules है, ठीक है. Next we discuss about pollination. See, एक flower से दूसरे flower में how it germinates. It can germinate either by wind, water, or through insects or birds. ठीक है, transfer pollen grains from anther to stigma of a pistil. It can occur through what? Through wind, water, or through biotic agents. Okay. And types of pollination में self pollination करके होता है and cross pollination करके होता है. We encourage cross pollination, that is called as xenogamy, and discourage self pollination. Self pollination, why we discourage, is because it will cause inbreeding depression. Self pollination के अंदर दो टाइप के हैं, autogamy and geitonogamy. और autogamy के अंदर दो टाइप्स है, that is chasmogamous and chastogamous. We will see self pollination में autogamy है. Autogamy में chasmogamous and clistogamous है. Chasmogamous is exposed anthers and stigma. It is more extrovert. Clistogamous मतलब it is more introvert. They do not open at all. They lie close to each other. ठीक है. और geitonogamy मतलब same plant में female और male होना and transfer of pollen grains from them. And xenogamy मतलब transfer of from one plant to another plant. इसमें क्या है, ऑटोगैमी में एक ही प्लांट में मेल और फीमेल होना. अभी एंड द नेक्स्ट वन इज गैमेटोगैमी में एक प्लांट में एक मेल और फीमेल क्रॉस जो पॉलिनेशन होता है उसको बोलते हैं गैमेटोगैमी. एंड xenogamy में कंप्लीटली डिफरेंट प्लांट्स, टू डिफरेंट प्लांट्स जो pollen grains ट्रांसफर होते हैं, दैट इज कॉल्ड एज xenogamy. ओके. एंड हाउ इट हैपेंस? इट हैपेंस थ्रू some agents such as water, wind or biotic agents. ठीक है. Wind, if the pollination occurs by wind, it is called as anemophily. ठीक है. It is quite common in grasses. Remember, if it is pollination occurring by water, it is called as hydrophily. ठीक है. इसमें इसका example Vallisneria and Hydrola. Remember, इसमें एक फीमेल क्या है, Vallisneria में क्या है, फीमेल फ्लावर reaches the surface of water by long stalk. फीमेल टॉल होते हैं और मेल कंपैरेटिवली छोटे होते हैं and releases the pollen grains on the surface of the water, and the water currents carry the pollen grains to reach up to the height of female flower. पहले स्टेज में फीमेल बड़ी है मेल से comparatively. And other example is sea grasses. Female flowers remain submerged in water, whereas pollen grains are long and ribbon-like. Pollen grains, जो male reproductive part है, that is taller than female. They are carried inside the water and reach the stigma. This is about pollination by water. And how it is not a wetty thing. ये जो pollen grains है, they are what protected from wetting. उसको क्या है, उसके पास क्या है, mucilaginous covering होते हैं, mucilaginous covering होते हैं. And pollination by insects is called as entomophily. Remember? And devices promoting cross-pollination. Unisexuality में monoecious dioecious है, dichogamy में क्या है, protandrous and protogenous है, then hercogamy, self-fertility, sterility, and heterostyling. These are what? Devices which promote cross-pollination. Understanding? Unisexuality में अभी इसमें क्या बोल रहे हैं? Why are we going for cross-pollination? Because self-pollination results in inbreeding depression. To avoid self-pollination and encourage cross-pollination, there are some devices in plants. First is avoiding synchronization. Synchronization मतलब सब कुछ same time में होना. So avoiding same time, which means what? इसमें छोटे ब्रैकेट्स में मैंने लिखा है डाइकोगैमी में टाइम लिखा है मैंने, ठीक है, डाइकोगैमी के नीचे टाइम लिखा है. और इसमें दो टाइप्स है, प्रोटोएंट्रस एंड प्रोटोगाइनस. प्रोटोएंट्रस में क्या होता है, एंट्रेशन, दैट मीन्स मेल रिप्रोडक्टिव पार्ट पहले आते हैं और फीमेल रिप्रोडक्टिव पार्ट लेट आते हैं. मतलब टाइमिंग हो गया, ठीक है, पहले मेल आता है और फीमेल आते हैं. एंड प्रोटोगाइनस में क्या है, पहले फीमेल आएगी, थोड़ा लेट मेल आएंगे, तो टाइमिंग का डिफरेंस दैट इज कॉल्ड एज डाइकोगैमी, ठीक है. एंड बी, arrangement of anther and stigma at different positions. So here position के बारे में बोल रहे हैं. इसमें क्या है, हेटेरोगैमी पोजीशन मैंने ब्रैकेट में लिखा है एंड हेटेरोस्टाइली ब्रैकेट में पोजीशन लिखा है, ठीक है. व्हाट इज हेटेरोगैमी? Heterogamy means there is a mechanical or a structural barrier is there, where in some flowers the pollen of a flower cannot reach the stigma of the same flower, ठीक है, because there is a hood or a covering. The stigma acts as a mechanical barrier, ठीक है, that is in heterogamy, एक mechanical barrier है. And in heterostyly, the stigma and anthers grow at different heights, which does not favor self-pollination. इसके example है primrose. Heterostyly के example है primrose, and hercogamy का example है pansy. Okay. And self-sterility. Sterile होना मतलब क्या होता है? Sterile होना मतलब prevent, it is like same, ज़्यादा improvements नहीं, कुछ भी नहीं. Okay, it is a mechanism to prevent self-pollen from fertilizing, inhibiting pollen germination. It inhibits pollen germination or pollen tube growth in pistil. Okay, that is called as self-incompatibility, नहीं तो self-sterility. And the last one, we are talking about what? Unisexuality. Unisexuality में क्या क्या है? Monoecious and dioecious. What do you mean by monoecious and dioecious? Monoecious मतलब examples क्या है? Castor and maize में where male and female flowers are present on the same flower. It is called as monoecious. Dioecious मतलब examples है papaya etc. where male and female flowers are present on different plants. Okay, monoecious and dioecious. Monoecious में क्या है? They prevent autogamy but not geitonogamy. Dioecious में क्या है? It prevents both autogamy and geitonogamy. Okay. ठीक है. Now we'll enter. अभी तक हम लोग रिविजन कर रहे थे. ठीक है. Do you want to take screenshot? You can take it now. Do you want me to go through any slides again? You can take screenshots. Okay. We are going to start with the new topic, fresh topic for today. Do you want to take screenshots? You tell, I will stop wherever you asked, wherever you want to take. Can I start with a new topic?
Yes.
Great. So here pollen and pistil interaction. Pollen मतलब male, pistil मतलब female interact कैसे होते हैं वो हम जानने वाले. Okay. So it is a process in which pistil recognizes compatible or incompatible pollen through chemical components produced by them. The girls, example girls सोचेंगे हाँ ये लड़का ठीक है, हाँ तो ठीक है चल जाएगा. अगर ठीक नहीं है, so that's not a compatible नहीं है, right type नहीं है, वो wrong type है, bad type है. ठीक है. So if the pollen is compatible, right type, the pistil accepts it. वो बंदा ठीक है, तो मैं accept करेगी. And promotes post-pollination events. ठीक है. Pollen grains germinates on stigma to produce a pollen tube. Pollen grains जब आते हैं stigma के ऊपर, it will germinate and it forms what? It produces a pollen tube through one of the germ pores. जब pollen grain आ जाते हैं stigma के ऊपर, it will produce pollen tube. The contents of the pollen grain move into the pollen tube. Pollen tube grows through the tissues of stigma and style and reaches the ovary. ठीक है? So if the pollen is incompatible, the pistil rejects the pollen by preventing pollen germination or the pollen tube growth. जब दोनों compatible है तो ही pollen tube growth होएगा, नहीं तो it won't germinate. Pollen germination will not happen or the pollen tube growth will not happen if it is not compatible. In plants which shed pollen grains at two-celled condition, two-celled में एक is vegetative and other is generative. And the generative cells divides into two male gametes during pollen tube growth. Okay.
Yes, sir.
And in plants which gives three-celled condition, pollen tubes carry two male gametes from the beginning. pollen tube reaches the ovary, then enters the ovule through where? Micropylar end, remember? उसमें एक gap रहता है. Through that only the male gametes enter. Micropylar end, chalazal end, remember? So micropyle से ही this pollen tube will reach the ovary, then enters the ovule through micropyle and then enters one of the synergids through the filiform apparatus. Filiform apparatus is like a finger-like apparatus, जो synergids में है. The filiform apparatus present at the micropylar part of the synergids guide the entry of the pollen tube. How it guides? It guides by releasing some chemical components that is called as chemotaxis. Okay. ये गाइड करते हैं कैसे, वो रास्ता दिखाएगा कैसे? By throwing some chemical components. और पोलन ट्यूब को समझ में आएगा, ओके ओके हाँ ये यहाँ है करके. तो in this way this will guide the entry of the pollen tube. This is the pollen pistil interaction. Now next topic is artificial hybridization. It is a crop improvement program in which desired pollen grains are used for pollination, and how it is achieved by emasculation and bagging. Emasculation matlab removal of anther. Anther matlab male reproductive part. Second is bagging. Bagging matlab you see the fourth picture. It is covered by with a bag or a cover. Okay. Now we'll discuss in detail. Artificial hybridization में emasculation. This is removal of anthers from bisexual flower bud of a female parent before the anther dehisces. मतलब before the anther bursts out, it will remove the anther from the flower. It is called as emasculation. Second one, bagging. Here, emasculated flowers, जो anthers निकाले हुए flowers are covered with a bag. किसका एंथस निकाला है उसको अभी कवर करना है, नहीं तो कुछ और उसके ऊपर आके बैठेगा. ठीक है. वी वांट अ डिजायर्ड पोलन ग्रेन. मुझे जो चाहिए पोलन ग्रेन में उसको वो ही लगाने वाला. आई डोंट वांट द एनवायरनमेंट, द विंड, वाटर और इंसेक्ट्स टू डू दैट थिंग. आई वांट माय डिजायर्ड पोलन ग्रेन टू बी अटैच्ड टू दैट. ठीक है. सो अ बैग इज कवर्ड विद इट to prevent contamination of its stigma with unwanted pollen. When the stigma attains receptivity, see आपके घर में कोई आएगा, you will enter the person when he or she you knew it. If the person is unknown, you can't accept him, right? You will allow. Do you allow him to enter the house? No. So when the when the stigma attains receptivity, mature pollen grains collected from anthers of the male parent are dusted onto the stigma. हमारा desired matured pollen grains I have already collected from the anthers and they are dusted onto the stigma. Then the flowers are rebagged. Then again it is rebagged. वापस इसको bag किया रहेगा, and which will allow them to develop into a fruit. For this is about, we talked about bisection. This for this. Now we are going to talk about unisexual. There's nothing much. There will be no need of emasculation क्योंकि वो एक ही either male or female. So female flower buds are bagged before flowers open. When the stigma becomes receptive, pollination is carried out using the desired pollen and the flower is again pre-bagged. ठीक है, as simple as that. Now we will talk about double fertilization. इसके पहले हम लोग ने artificial hybridization किया है और उससे भी पहले we talked about pistil and pollen pistil interaction. Now we will talk about double fertilization. So after entering one of the synergids, the pollen tube releases the two male gametes into the cytoplasm of synergid. One male gamete moves towards the egg cell and fuses with its nucleus, syngamy bolte hai, to form a zygote. Another male gamete moves towards the two polar nuclei located in the central cell and forms with them to form a triploid primary endosperm nucleus, PEN. As it involves fusion of three haploid nuclei, it is called as triple fusion. इसमें क्या है पिक्चर आपको क्या बताऊं हाँ पहला का I will show you this picture. Here, two male gametes. MG दिख रहा है male gametes? दो है. ये pollen tube से आ रहे हैं and it will enter over here. ठीक है. So one male gamete will fuse with the egg. Egg दिख रहा है आपको? Female में egg दिख रहा है? So one male gamete will form with the egg and this will further form what? Zygote. ठीक है. And the second male gamete जो है जो दूसरा है that will go where? It will go to the central area. ये जो polar nuclei है वहां जाएगा and forms what? PEN. यहाँ first male gamete will go and binds to egg and it will form zygote, and the second male gamete go and attaches to the polar nuclei and form PEN. What is PEN? अब भी रीड किया था. What is PEN? Primary endosperm nucleus.
Nucleus.
So this will what? Form triple fusion. ये zygote होएगा, that is diploid, and this is triple fusion, PEN. Understood? The
Yes.
pollen tube releases two male gametes into the cytoplasm of the synergid. One male gamete moves towards the egg cell and fuses with its nucleus to form what? Zygote. That is a diploid. And other male gamete move towards the two polar nuclei located in the central cell and forms to produce a triploid primary endosperm nucleus, PEN. Great. Good enough? Since two types of fusion, that is syngamy, which is with the egg, and triple fusion, which is with the central nuclei, takes place in an embryo sac, it is called as what? Double fertilization. It is an event unique to flowering plants. The central cell after triple fusion becomes PEC, primary endosperm cell, and develops into endosperm. Then zygote develops into an embryo. पहला क्या है जो ये है PEN थे. Then it will form PEC. Then it develops into endosperm and later into an embryo. ठीक है. पहला क्या होगा? एक मेल गैमेट एक के साथ फ्यूज होगा टू फॉर्म अ जाइगोट. जाइगोट कहाँ है? माइक्रोपाइलर एंड में है. ये देखो यहाँ है. यू कैन सी वेयर आई एम पॉइंटिंग.
Yes, ma'am.
sorry, not over there. Here. Extremely sorry. जाइगोट यहाँ है. ठीक है. ये पीछे थोड़ा उल्टा किया उन लोग ने. ठीक है. सो इट इज द माइक्रोपाइलर एंड. तो यहाँ रहेगा क्योंकि सिनर्जिड्स यहाँ है तो उसका सीधा ऑपोजिट क्या है? एंटीपोडल सेल्स रहेंगे. ठीक है. पिक्चर लोगों ने थोड़ा सा उल्टा किया. ठीक है. सो माइक्रोपाइलर ट्यूब यहाँ से आएंगे. यहाँ दो मेल गैमेट रहेंगे. एक एग के साथ फ्यूज होगा टू फॉर्म जाइगोट एंड दूसरा मेल गैमेट इट विल फ्यूज विद द पोलर न्यूक्लिया. Okay, to form what? PEN. Then PEN develops into PEC, that is primary endosperm cell. and further PEC develops into what? Endosperm हो जाता है, ठीक है, एंडोस्पर्म होएगा and last में एंडोस्पर्म क्या होएगा? Embryo, zygote develops into an embryo. Zygote will develop into a embryo. PEN, PEC, endosperm. Okay. Zygote यहाँ जो है माइक्रोपाइलर एंड में है. zygote उसके बाद क्या होएगा? After fertilization, it will form what? Embryo. We will learn about them in later slides. So here PEN, PEC, endosperm हो जाएंगे and zygote will form after fertilization into an embryo. Okay. So this is about fertilization. ये fertilization के बारे में बोल रहा है. And after fertilization के बारे में see parts before fertilization and transformation after fertilization. जो भी हम लोग ने देखा sequence, petals, stamen, style, sigma, ठीक है, before fertilization, वो usually after fertilization wither, नहीं तो fall off. It will either fall off or wither. मतलब ड्राई होके चले जाएंगे. Ovary will transform into a fruit, ovule into a seed, egg into zygote. You saw egg will fuse with one male gamete, then forms what? Zygote, isn't it? Funicle to stalk of the seed. Micropyle will be micropyle. उसको कोई change नहीं है. But it will facilitate what? Oxygen and water uptake. पानी चाहिए ना जीने के लिए, ऑक्सीजन चाहिए ना. Then nucellus. Nucellus means what? It is a parenchymatic cell, which means what? Food reserve. They will produce, they will make food. वो nucellus क्या होएगा? It will form perisperm. हमारा if you see we humans, we will be a baby first. तो baby को हम लोग baby बुलाएंगे. उसके बाद will become a child. Child से teenage. Teenage से adulthood. Then senior citizen. ठीक है, एक एक नाम है ना. When we grow, we are given a specific name. Humans are given a specific name. Similarly, for plants, as simple as that. An outer integument, the covering, is called as testa. And inner integuments. Integument में दो type है, outer and inner. वो जो पहले outer थे, वो अभी क्या होएंगे after fertilization? Testa बोलेंगे. और integument, inner integument को क्या बोलेंगे? Tegmen बोलेंगे. Okay. Synergid cell will degenerate. Secondary nucleus जो है वो endosperm होएगा. I said about PEN will form PEC, then it will form endosperm. है वो बड़ा बड़ा होते जाएगा, उसका नाम भी चेंज होते जाएगा. Similarly zygote वो बड़ा होते जाएगा, तो after fertilization will name as embryo. Zygote will form what? Embryo. Endosperm होएगा कभी? ये जो two nucleus cell will form into endosperm. उसके बीच का प्रोसेस है PEN and PEC. ठीक है. Antipodal cell it will degenerate. क्या क्या degenerate होएगा? Antipodal cells degenerate होएंगे. Synergid cell degenerate होएंगे. Synergid कौन सा है? दो synergid cells है. Finger like structure. Fibril apparatus. वो degenerate होएंगे. और क्या degenerate हो रहा है इसमें? Sepals, petals, stamen, style and stigma will fall off, will wither off. ठीक है. And next is post fertilization. These are हम लोग ने जो भी अभी तक सीखा है, sepals, petals, stigma, everything. Ovule, egg, funicle, nucellus, integuments, synergid, antipodal. These are parts before fertilization. Now we are going to talk about post-fertilization structures. Post-fertilization events. क्या क्या है post-fertilization events? Endosperm होएगा. PEN, PEC, then endosperm, remember? Embryo develop होएगा कैसे? Zygote से embryo बोला था. Okay. Ovule into seed, ovary into fruit. यहाँ लिखा है. Ovule into seed, ovary into fruit. Okay, these are the major changes will happen post fertilization of a flowering plant. So first one is endosperm development. We will discuss in detail. Primary endosperm cell (PEC) divides repeatedly to form a triploid endosperm tissue. Endosperm cells are filled with what? Endosperm का use क्या है? They make food. They reserve food materials. They are used for nutrition for the developing embryo. Developing embryo के लिए लोग जो है एंडोस्पर्म है वो बीच में है, ठीक है. वो PEN, PEC उसके बाद एंडोस्पर्म होता है, वो बड़ा बड़ा होते जा रहा है. वो क्यों बड़ा हो रहा है? It reserves food materials. Why? To nourish the embryo, developing embryo. Zygote से भी embryo बनेगा, उसको food चाहिए ना, तो it will reserve food for the embryo. In common endosperm development, PEN undergoes successive nuclear division to give free nuclear. This stage is called as free nuclear endosperm. Okay, now we'll say about free nuclear endosperm. So endosperm में types है. There are different types of endosperm. That is nuclear endosperm, cellular endosperm, and halobial endosperm. So do you have any idea what is it? Nuclear. Nuclear मतलब सिर्फ nucleus ही है, खुला खुला ऐसे ही बहुत सारा बनाते रहते हैं nuclear. Free nuclear बना रहा है. Cellular endosperm मतलब cells भी बना रहे हैं और साथ ही साथ nucleus भी बना रहे हैं. See, two circles are there, one big circle, small circle. It forms cell also. Along with it, it forms nucleus also. That is called as cellular endosperm. And nuclear endosperm में क्या है? Free nuclear बना है. See, only nucleus. Nucleus is there. And halobial. Halobial means what? It is somewhere something between nuclear and cellular. Okay. So in nuclear endosperm, the primary endosperm nucleus divides by mitotic division. ठीक है? Mitotic free nuclear divisions without the formation of walls. इसके examples क्या क्या है? Coconut water है. Coconut जो है coconut, cotton, and in cellular endosperm, the first nuclear division of the primary endosperm nucleus is immediately followed by a wall formation. Example, petunia flower, datura. Okay. इसमें wall नहीं है, cell wall नहीं है, इसमें cell wall है. And in halobial, it is an intermediate type of endosperm formation between a cellular and a nuclear type of endosperm. Halobial type of endosperm development is prevalent in monocotyledons. ये monocotyledons में ज़्यादा दिखता है, halobial type of endosperm. So this diagram, so first we'll finish this. So in common endosperm development, PEN undergoes successive nuclear divisions to give free nuclear. This stage is called as free nuclear endosperm. That means they're talking about this free nuclear. Number of free nuclei varies greatly. Endosperm becomes cellular due to cell wall formation. Endosperm becomes cellular due to cell wall formation. So it is called this. They are talking about cellular formation. And they are not much talking about this, the third type. And tender coconut water is a free nuclear endosperm made up of thousands of nuclei, and the surrounding white kernel, ये जो है, is the cellular endosperm. So this is the nuclear endosperm. जो पानी है nuclear endosperm. This is the cellular endosperm. Great enough understanding?
Yes.
Great. Okay. So I just wanted to show this. Angiosperms मतलब flowering plant. ठीक है. Flowering plant are always 3N. तो 3N कहाँ से मिलते हैं? Triploid है. 3N कहाँ से मिलते हैं? 2N from its mother and 1N from the father. And it forms 3N, that is triploid. Gymnosperms such as spinous, cycles, which we have studied in 11th standard. Remember the first or second chapter, botany में. So it is always haploid, N है. Only single N it has, and it is from mother. Mother gives, mother हमेशा carrying रहता है, and father gives 0. So N plus 0 becomes N, haploid. Gymnosperm के N है, N haploid है. Angiosperms के 3N है, that is triploid. This was just a pictorial representation, that's it. I guess I pasted in the wrong slide, I guess. It should be mentioned prior to this slide. Okay, so we have talked about endosperm development and types of endosperm, that is nuclear, cellular, and halobial. Now we will talk about embryo development. Okay, post fertilization में क्या क्या है? Endosperm, embryo, ovule into seed, and ovary into fruit. We finish this, we'll go to embryo development. Embryo develops at the micropylar end. This is a nuclear, central nuclear area. This is the zygote area. Zygote will form into embryo. Where? Which end it is? Micropylar end. Micropylar end कहाँ क्या आएगा? Pollen tube रहेगा. कहाँ यहाँ से क्या आता है? Male gametes आते हैं. This is the micropylar के opposite end को क्या बोलते हैं? Chalaza end. Chalaza end में क्या क्या antipodal cells है वो मगर fertilization में degenerate होते हैं. See, it is mentioned as degenerating antipodal cell. ठीक है, so embryo develops at the micropylar end of the embryo sac where the zygote is situated. Most zygotes divide only after the formation of some endosperm. This gives nutrition to the developing embryo. Endosperm makes food. It will provide some nourishment to the embryo, the developing embryo. In monocots and dicots, seeds differ greatly, but embryogeny, that is early embryonic development, is similar. ये जो है हम जो बात कर रहे हैं zygote से लेके embryo तक जो developing age है, this is the stage is similar in both monocots and dicots. This is what they are saying. And embryo development में क्या क्या है? Zygote से pro embryo होएगा. Pro embryo से globular, then heart shape, then mature embryo. So zygote is over here, the second picture over here. ठीक है, it will flow to pro embryo, it will develop into pro embryo. और उससे भी थोड़ा बड़ा होएगा, that is globular embryo. Globular embryo देखो global जैसा round figure है, see? And this, when it forms a heart shape and globular round shape से heart shape में होता है, that is called a heart shape embryo, see? And further it will form into a mature embryo. ठीक है? This is embryo development, embryogeny. So embryo किस तरह के है? Dicot and monocot. It has an embryonal axis and two cotyledons. ठीक है. Embryo development में dicotyledon और monocotyledon है. So in the portion of embryonal axis above the level. So here in this picture, a typical dicot embryo. So seed say, now we're going to, this is dicot. How we will come to know it is a dicot? एक डंडा दिख रहा है, दूसरा डंडा दिख रहा है. This is one, this is two. So two dicots. So one dicot, sorry. So this is a dicotyledon. और इसके डाइकॉट जो कॉटलिडॉन्स के ऊपर के जो एरिया को बोलते हैं एपिकॉटिल. एपी मतलब ऑन द सरफेस ऑफ, ऑन द सरफेस बोलते हैं. सो दैट इज एपिकॉटिल. एपी मतलब ऑन टॉप ऑफ द सरफेस. सो कॉटलिडॉन के ऊपर जो है एपिकॉटिल बोलते हैं. कॉटलिडॉन के नीचे जो है, अंदर जो है, दैट इज कॉल्ड एज हाइपोकॉटिल. ठीक है. एंड रेडिकल करके एक चीज है, प्लूम्यूल करके एक चीज है. प्लूम्यूल से क्या बनता है? शूट बनते हैं. इट विल स्टार्ट टू फॉर्म शूट अपेक्स फ्रॉम प्लूम्यूल. एंड रेडिकल से क्या बनता है? इट विल स्टार्ट टू फॉर्म रूट. ठीक है. रेडिकल से रूट होते हैं, प्लूम्यूल से शूट होते हैं. तो कॉटिलेड दे प्रोसेस ओनली, oh god, where is the picture? Dicot. So it has an embryonal axis which includes plumule से लेके root cap तक and two cotyledons. That is two cotyledons. The portion of embryonal axis above the level of cotyledon is the epicotyl. The portion of embryonal axis above the level of cotyledon is called as epicotyl. Cotyledon के ऊपर जो है epicotyl, cotyledon के नीचे जो है hypocotyl. Then epicotyl which terminates with the plumule, stem के tip पे. The cylindrical portion below the level of cotyledon is hypocotyl that terminates with a radicle. Epicotyl से plumule, hypocotyl से radicle, root tip. The root tip is covered with a root cap. The root tip is covered with a root cap. Radicle को root tip भी बोलते हैं और radicle और root tip is covered with a root cap. एक capping दिया गया है. In the next slide, we are talking about monocotyledons. So what is the different thing from dicot? Monocot में क्या different है? As we will see, they possess only one cotyledon. And उसको एक cotyledon को क्या बोलते हैं? Scutellum. Cotyledon of the grass family is called scutellum. Any monocot नहीं तो cotyledon is called as scutellum. इसको cotyledon नहीं तो scutellum भी बोलते हैं. That is this. It is situated laterally to the embryonal axis. At its lower end, the embryonal axis has the radicle and the root cap enclosed in coleorhiza. ठीक है. Radicle है which forms into root. और उसका root cap ही है. और root cap is enclosed in coleorhiza. Root cap enclosed in coleorhiza. This is the extra thing in monocotyledon. And next, the other extra thing, the portion of embryonal axis above the level of attachment of scutellum, which means cotyledon, is the epicotyl. ठीक है. cotyledon के ऊपर की चीज को क्या बोलते हैं? एपिकॉटिल. cotyledon को दूसरा नाम है scutellum. scutellum के ऊपर के चीज का नाम क्या होते हैं? एपिकॉटिल. और एपिकॉटिल में क्या है? इट हैज shoot apex and a few leaf primordia enclosed in coleoptile. coleoptile मतलब it has shoot apex and a few leaf enclosed in coleoptile. ठीक है. So इनमें exact coleorhiza and coleoptile है. And what else is there? Do you see something different? There is a epiblast. What is epiblast? When the second cotyledon degenerates, उसका जो बाकी बचा हुआ है दैट इज कॉल्ड एज एपीब्लास्ट। दिस इज मोनोकोटिलीडॉन। मोनोकोटिल में से एक ही कॉटिलीडॉन है, मगर दूसरा कॉटिलीडॉन जब डिजेनरेट होते हैं तो बच्चा कुछ चीज जो होते हैं उसको बोलते हैं एपीब्लास्ट। सो फ्रॉम डाइकॉट व्हाट इज डिफरेंट इन मोनोकोट? 3 थिंग्स एक्स्ट्रा है, दैट इज कोलोराइजा, एपीब्लास्ट एंड कोलियोप्टेरा। Radicle is what? The root of the embryo. Okay. Plumule मतलब क्या है डाइकॉट में? What is plumule? It will generate into what? Shoot. Shoot मतलब छोटे छोटे लीव्स आने लगेंगे यहाँ से, प्लूम्यूल से। प्लूम्यूल से छोटे छोटे लीफ आने लगेंगे, शूट अपेक्स। रेडिकल मतलब रूट, रूट आने लगेंगे रेडिकल से। ठीक है, अंडरस्टैंडिंग? Yes, class, understanding?
Yes, ma'am. Yes.
Okay, okay, okay. Okay, so this was embryo development. This is we are talking about post fertilization में endosperm development दिखे and embryo development देखा अभी। और embryo development में monocotyledon, dicotyledon और उसके shape क्या क्या है, zygote to mature embryo के बीच में क्या क्या है, pro embryo है, globular है, heart shaped है, then it forms a mature embryo. Endosperm में हम लोग ने क्या क्या देखा? Types of endosperm, nuclear, सेल्यूलर, हेलोबियल। न्यूक्लियर मतलब फ्री रेडिकल। कोकोनट वाटर जो है दैट इज न्यूक्लियर विदाउट एनी सेल वॉल। सेल्यूलर मतलब इट विल हैव अ सेल एंड इनसाइड दैट इट विल हैव अ न्यूक्लियस। ठीक है, और कोकोनट वाटर के जो वाटर में उसके बाहर का जो व्हाइट कर्नल होता है दैट इज अ सेल्यूलर एंडोस्पर्म। एंड हेलोबियल इज समथिंग डिफरेंट व्हिच इज an intermediate of nuclear and cellular type. And the next one is seed from ovule. Post fertilization में तीसरा चीज है seed from ovule. So seed is the fertilized ovule from formed inside fruit. फ्रूट के अंदर जो होते हैं fertilized ovule को क्या बोलते हैं? सीड बोलते हैं. It is a final product of sexual reproduction. It is a final product of sexual reproduction. It consists of seed coat, cotyledon or cotyledons and embryo axis. सीड कोट करके एक्स्ट्रा चीज है, सीड है तो उसको एक कोटिंग भी रहेगा, और cotyledon और embryo axis just like monocot and dicot we studied. The cotyledons are simple, generally thick and swollen due to storage food, as in legumes and all. ठीक है, you can see this figure. Dicot seed में क्या है? सीड कोटिंग है, हाइपोकॉटिल है, एपिकॉटिल है। एपी मतलब कॉटिलिडन के ऊपर जो है एपी, और कॉटिलिडन के नीचे जो है हाइपोकॉटिलिडन। ठीक है, एंड दिस इज मोनोकॉटिलिडन सीड। पहला आउटर कवरिंग विल बी अ सीड कोट, देन एंडोस्पर्म। एंडोस्पर्म क्या करता है? फूड बनाता है। एंड एंड दिस इज द कॉटिलिडन, और उसके ऊपर का चीज को क्या बोलते हैं? एपिकॉटिलिडन। कॉटिलिडन के नीचे का क्या बोलते हैं? हाइपोकॉटिलिडन। रेडिकल किसके लिए? फॉर इट इज अ रूट ऑफ द embryo. So mature seeds are of two types, non-albuminous and albuminous. Non-albuminous मतलब no residual endosperm. मतलब there is no खाना making as it is completely consumed during embryo development. There is no bacha kucha food and which is completely consumed in embryo development. Example pea, groundnut, beans. ये जो है they are non-albuminous. There is no residue. कुछ भी बचा नहीं है. Everything is consumed. Albuminous seed. They retain some endosperm. मतलब कुछ तो खाना बचा है. That is example क्या है? Wheat, maize, barley, castor, coconut, sunflower. Okay, seeds are of, mature seeds are of two types, albuminous and non-albuminous. Albuminous matlab they retain some endosperm. Non-albuminous matlab they don't retain endosperm. It is completely consumed during embryo development. Occasionally in some seeds such as black pepper and beet, the remnants of nucellus are also persistent. It is called as perisperm. Sometimes in some seeds, there will be something remaining from the nucellus. Okay, and that something which is remaining is called as perisperm. Perisperm हम लोगों ने कहाँ देखा था? In the very first diagram, it was saying nucellus will form after fertilization. Perisperm हो जाता है, and something बचा कुचा रहेगा if it persists. ठीक है, nucellus अगर existing है तो after fertilization will form. It is called as perisperm. ठीक है. So integuments of ovules. Embryo sac में तो integuments से covering से inner integument and outer integument. So integuments of ovules harden has tough protective seed coats. Integument before fertilization it is called as integuments, covering, outer covering. उसके बाद क्या बोलते हैं? Protective seed coats बोलते हैं. It has a small pore that is a micropyle. Micropyle is still the same. After fertilization, it is called as micropyle only. So what it does? The micropyle क्या है? Pollen tube के साथ attach होएंगे, isn't it? Why it attaches? It attaches for what purpose? Oxygen intake and water intake into the seed during germination process. Okay. And as the seed mature, it becomes dry by reducing water content. जब mature हो जाता है तो इसका water content कम होते जाएगा. The metabolic activity of embryos slows down and it enters a state of dormancy or inactivity. Under favorable condition, they germinate. Favorable conditions क्या है? Moisture, oxygen, and temperature. What are the advantages of seeds? So since pollination and fertilization are independent of water, seed formation is more dependable. मतलब उसको पानी चाहिए रहता है. Better adaptive strategies for dispersal to new habitat. It helps the species to colonize in other areas. They have food reserves. Food है उनके पास. They can make food. So young seedlings are nourished until they are capable of photosynthesis. ठीक है, young seedlings. They are given food until they are capable of photosynthesis. The hard seed coat protects the young embryo. They generate into genetic combinations and variations. Dehydrations and dormancy of mature seeds are crucial for storage of seed. It can be used as food throughout the year and also to raise crop in the next season. We can store them and we can raise crop in the next season. So what the next is viability of seeds after dispersal. So what is viability? It means how long you can live, how long it is viable. So in few species, the seeds lose viability within a month, within few months. Seeds of many species live for several years also. Okay, some for few months and other for some years also. Some seeds that remain alive for hundreds of years. The oldest is that of a lupine, Lupinus arcticus, excavated from Arctic tundra. The seed germinated and flowered after an estimated record of 10,000 years of dormancy, inactivity. See, मतलब वो जिंदा थे, but it was inactive. 2,000-year-old viable seed is of the date palm, which is called as Phoenix dactylifera, discovered during the archaeological excavation at King Herod's palace near the Dead Sea. फिनिक्स मतलब क्या होता है? मर के वापस जिंदा होना, फिनिक्स सीड, लुपिनस सीड. ठीक है, लुपिनस आर्टिकस एंड फिनिक्स मींस डेट के सीड, डेट पाम. ओके, एडवांटेजेस हम लोग ने भी saw advantages. And what else? Types of seed. Okay, advantages, types of seed, and viability of seed after dispersal. For example, lupine और date palm के. So seed का इतना हमको बताना पड़ेगा. सीड के बारे में, सीड डेवलपमेंट के बारे में बोलेंगे तो क्या क्या बताएंगे, टाइप्स क्या क्या है, और उसके वायबिलिटी आफ्टर डिस्पर्सल क्या है, एंड एडवांटेजेस क्या क्या, 6 एडवांटेजेस है वो भी बताना है. ठीक है. And next is fruit. The fourth one, post fertilization, is a fruit from ovary. So the ovary develops into a fruit. Ovule is developed into a what? Ovule. Correct. Ovule is developed into a seed and ovary develops into a fruit. Transformation of ovules into seed and ovary into fruit proceeds simultaneously. The wall of ovary develops into pericarp. Ovary की जो ovary wall है, it will form pericarp, wall of the fruit. The fruits may be fleshy or dry. Fleshy क्या क्या है? Examples guava है, orange, mango. Dry क्या क्या है? Examples groundnut है, mustard है. Okay. So there are two types of fruits: true fruits and false fruit. If the fruit develops only from the ovary, जो usual process है, उसको क्या बोलते हैं? True fruits. Other floral part degenerate and fall off. Example क्या क्या रहेगा? Mango है, grapes है, like mostly all plants. Okay, so that is about true fruit. False fruit क्या है? When the fruit develops from the thalamus, this is called as false fruit. Examples क्या क्या है? Apple, strawberry, cashew nut. Apple, strawberry, cashew nut. सीड्स के दो टाइप होते हैं, फ्रूट के भी दो टाइप है, दैट इज ट्रू फ्रूट एंड फॉल्स फ्रूट. In some species, fruits develop without fertilization. Such fruits are called as what? Parthenocarpic fruits. Example banana है. Parthenocarpy can be induced through the application of growth hormones. Such fruits are seedless. अभी नए नए मार्केट में नया आया है seedless papaya. I have got such things. Seedless watermelon. In some places, not in all places, in some places. So how they are induced through a growth hormone. और वो ये जो प्रोसेस को क्या बोलते हैं? Parthenocarpy. Parthenocarpy, that means what? They are fruits developed without fertilization. They don't have seed. They are seedless. This is called as parthenocarpy. ठीक है. So this is for today's class. I hope you guys understood something. Do you want me to wait for you to get the screenshot? Any slides you want to get the screenshot? Please tell me.
Ma'am, can you send this whole PDF on the group, official group?
Okay, okay, okay. And in that group, it includes all people, right? तुम लोग 15
Yes, ma'am. Teachers and students.
Okay, and if I want to connect only to you people, only to biology students, PCMB भी है ना, मैथ्स के बच्चे भी है ना उसमें.
Yes, ma'am.
So how will I? Is there any other group only of?
No, ma'am. We have only one group.
I see. Okay, okay. ठीक है. I will send across this. Okay then. So, do you want me to repeat anything? Yeah, we still have time. And we have only small portion left for this particular chapter to finish. That we'll take it in the next class and we'll revise it again. क्या बताया था? We'll revise it again so that it will be thorough in your mind. These are very, very simple
Yes.
if you see in the PPT and all, the PDF and all. But when you read the textbook, it is little complicated. So side by side, simultaneously, ये भी open करना और textbook भी open करके रखो. Then only you'll understand.
Yes, ma'am.
Yeah. So you want me to repeat anything? Just tell me anything for today's class or like previous class, anything. If no, then we can end the lecture.
Yes, ma'am.
Okay. Then please do revise, study whatever is there in the textbook for this chapter. Okay, I will send across the PDF today itself.
Yes,
Okay.
ma'am. Thank you, ma'am. Bye, ma'am.
Thank you. Thank you. Thank you so much.